Okay, I think we can start. Hi, everyone. My name is Rinat, and I am the Vice Chair of the Division of the Health and Nutrition Division at the AOCS. Welcome to this webinar. We are really excited to host this seminar and also to share with you the exciting research that you will hear about just in a bit. We have two distinguished speakers, Dr. Metherell from University of Toronto and Dr. Comberford from Nature Crops. They will introduce themselves just before the talk, so stay tuned. Maybe just one comment on housekeeping. You will see the question and answer box. Please use the Q&A in order to capture your comments, to capture your questions. We will look at them and we will discuss them after the two talks are finished, so at the end of the webinar. But please use it during the webinar. And I hope you will enjoy. We're really happy to have you here. And Dr. Metherell, I think the floor is yours.
Thank you, Rinat. My name is Adam Metherell. I'm an assistant professor at the University of Toronto in Canada. Quick little background, I did my postdoctoral fellowship with Richard Bazinet, also at the University of Toronto, and my graduate studies with Ken Stark at the University of Waterloo. I imagine that those are a couple of names that many of you might be familiar with. So today, I'm going to talk about compound-specific isotope analysis as an emerging tool for studying dietary intake and the metabolism of polyunsaturated fatty acids, specifically with a focus on omega-3 polyunsaturated fatty acids today. But just before we start, I'd like to acknowledge the land in which University of Toronto operates. For thousands of years, it's been the traditional land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. This meeting place is still home to many Indigenous people from across Turtle Island, and we're grateful to have the opportunity to work on this land. So just a little overview of what I'm going to talk about. I have the N3 PUFA biosynthesis pathway on the right. I'll talk a little more detail about that later. But I'll go over a little background on what compound-specific isotope analysis is, particularly with relation to natural abundance. And like I said, go over the N3 PUFA or polyunsaturated fatty acid biosynthesis pathway, and then touch on some new discoveries that have about this pathway that have been enabled by CSIA itself, particularly addressing whether DHA synthesis rates are low and how plant N3 PUFAs could be a good source of DHA. So just to touch now on what natural abundance compound-specific isotope analysis is, this natural abundance idea really comes down to the metabolism of plants, in particular the photosynthetic pathway for carbon fixation. Generally, most plants are categorized into an either C3 or C4 photosynthetic pathways. As a result of this, we actually get very distinct carbon-13 signatures depending on which pathway a plant uses. Many of us were taught that the amount of C13 that's present in the environment is about 1.1% of total carbon, where in fact it is actually a range. We get a range of about 0.96 to 1.15% of total carbon being C13, and this is very much dependent on where that's been derived from relative to the photosynthetic pathway. So there's a C3 pathway here, and I'll just kind of guide you a little bit here. The nomenclature for this is really quite cumbersome, but the C3 pathway discriminates against C13 molecules, so it tends to want to use C12 molecules to fixate carbons into glucose, into lipids, etc. So it has a lower carbon-13 content and a more negative miliary value, whereas the C4 pathway doesn't discriminate so much against C13, and that results in a higher C13 content. When it comes to plants, the majority of plant species are from the C3 pathway. Something over 90% of all plant species are the C3 pathway. Example here is a flax plant. But the C4 pathway, although it's much less common on a per species basis, we do have two particular plants, corn and sugarcane, that happen to use this C4 pathway. So these, of course, are very abundant in the food supply. And then with seafood and fish sources, this tends to be sort of a mixture and falls in a more intermediate range of C13 content. So this variation from the plants that are consumed by mammals, and then potentially those mammals being consumed by us and other animals, these can be detected and can really minimize the need for expensive tracers. And this is detected through something known as isotope ratio mass spectrometry, which can be coupled, especially in the case of fatty acid work, to gas chromatography to measure the isotopic signature of specific fatty acids. So what is isotope ratio mass spec? Well, essentially here we have what's showing as a gas chromatograph. Fatty acid methyl esters are injected into the column, just like a normal GCFID. Fatty acids separated by chain length desaturation. And then they exit the GC column and then go into a combustion or oxidation reactor. At this point, the oxygen combined with extremely high heat and the fatty acid methyl esters as a fuel, these are all combusted to form CO2 and water. The water is dried off in a dryer, and the CO2 enters into the mass spec, where there's some very specific detectors designed to analyze just each detector essentially analyzes one mass. In total, we analyze 3 masses, with the 44 mass to charge ratio being the C12 CO2 and the 45 being C13 CO2. And this ratio of C13 to C12 then is used to measure differences in natural abundance. And this is really a very sensitive, because the detector is so specific to certain masses, we actually can get very high sensitivity. When I mentioned 1.1% on average of all carbon is C13, we can actually detect variations to as little as essentially 1.111%. Of course, depending on sample sizes and that sort of thing. There's been some really neat applications of isotope ratio mass spectrometry that you may or may not be aware of. One really interesting one is with honey adulteration. So if companies are faking the sources of their honey, they could be adding high fructose corn syrup or sugarcane to it. That can be detected through isotope ratio mass spec. Blood doping, differences in endogenous or exogenous sources of certain metabolites can be detected. And one other really neat one, somewhat recently has been published, is food crusts from 6,000-year-old pots have been used to, isotope ratio used to identify the source of the food that was being consumed during the early Neolithic time period. So some really neat applications. For us, of course, none of them are as neat as looking at polyunsaturated fatty acid biosynthesis. But to go over this a little bit, this is the N3 PUFA biosynthesis pathway with ALA or 18,3N3 here as the essential omega-3 PUFA fatty acid. Through a series of elongation and desaturation steps, it can be converted to EPA or eicosapentaenoic acid here, and then eventually DHA or docosahexaenoic acid. And these are the two fatty acids we find primarily from fish and seafood, whereas ALA we get from plant and vegetable and seed sources, particularly high in chia and flax, but in our diet, we get it a lot from soybean or canola oil. So there's been a number of different dogma that's persisted throughout the years in relation to this pathway, despite the steps being fairly well known. There has been some ongoing debate. One of these dogma being that retroconversion can explain the increase in EPA when supplementing DHA. And this occurs in the peroxisomes, and we've actually shown very recently and with a recent article submission that DHA does not increase retroconversion. It actually prevents the synthesis of itself by inhibiting EPA metabolism. At the same time, there's this long time belief that the DHA synthesis rates are low. And we've shown a little bit that that may not be the case. And this is where I'm going to focus some of my results and research on now. So previously, there's been a number of methods used to assess DHA synthesis. The simplest being simple supplementation studies where you might supplement ALA and then measure changes or lack of changes in downstream metabolic products such as DHA. There are some infusion studies where you infuse a labeled precursor, again, such as a deuterated ALA, and measure the conversion of that label to DHA. And this has primarily been done in rodents with one fairly small study being performed in humans. And oral dosing studies, this is by far the most prominent isotopic type study in humans, where you provide a single dose of the labeled precursor and measure the accumulation of this, again, in downstream products, generally over days or perhaps weeks. So with regards to supplementation studies, it's fairly well established, the response to ALA supplementation, but here we have the change in blood ALA on the y-axis and the amount of ALA supplemented in a review paper. And you can see up to 14 grams of ALA per day increases blood ALA levels, increases blood EPA levels, but no change in DHA levels. And this is the type of study that has often been used as evidence for the idea that we are poor DHA synthesizers. Similarly, even supplementing EPA, we can see a large increase in EPA, DPA in 3, and again, no change in DHA. So once again, providing support for this idea that we are not very good at making our own DHA. So to summarize those, up to 14 grams ALA or 4 grams per day of EPA does not change plasma DHA levels. And really there's two plausible explanations for this. One is that very little precursor is being converted to DHA. That's kind of been the general belief. The other option is that what is being converted to DHA is being matched by downstream metabolism. So beta oxidation of DHA, conversion to oxylipins, endocannabinoids, and that type of thing. And we actually, just a few years ago, published an AJCN and very clearly showed that the second option here is really what's happening. We were able to use compound-specific isotope analysis to reveal that there was substantial conversion of EPA to DHA despite no changes in DHA levels. So the signature of the DHA in human blood was starting to match that of the EPA in the supplement. Now, switching to single oral dose studies, generally we have this one dose of ALA. It gets absorbed or digested, absorbed, transferred to sent out to tissues. Much of it going to adipose, a lot of it going to liver to be converted to products, and certainly beta oxidation being involved as well. There's been a number of different reports of percent of dose, percent of total plasma, but all of these have generally come to the consensus that conversion of dietary ALA to DHA is less than, or to plasma DHA specifically is less than 1% of that dietary ALA. There are a number of limitations. It tends to not take into account C13 ALA being stored in adipose that's later available for synthesis. It's also only measuring plasma. And of course, this is the limitation with any human work. Plasma and blood is certainly the easiest to measure, but it still isn't taking account into any DHA that's been synthesized and then taken up into other tissues. So I want to really highlight these 2 points. Remember this idea that we convert less than 1% of our dietary ALA to DHA, and that this is based on measurements of plasma only. So that's just to remember for later on in my talk. So getting into now this new, not new technology, but maybe new application of an old technology where we're using compound-specific isotope analysis and IRMS to perform these dietary and metabolic studies. So during my postdoc, we developed this diet switch model for determining N3 PUFA synthesis rates. And essentially what we do, we have the C13 signature on the y-axis over time. We feed the diet of a particular C13 signature. And then once they've kind of normalized to that diet and signature, we switch them to another diet of a different signature. And this can go either high signature to low, like this is showing, or it can be the opposite, low to high. And then after that diet switch, we can track the change in the signature in a variety of tissues under a variety of conditions over time. So we plot this tissue response over time, calculate the signatures, then we can use the initial starting point for the signature and the projected ending point or the plateau of the curve generated through one phase exponential decay with this equation. We can calculate half-lives based on the rate constant, and these half-lives can then be used to determine synthesis and or turnover rates by correcting for the levels of DHA in the tissue, for example. So this is not without its complications, particularly when we're talking about N3 PUFA metabolic studies. So the big issue here is that there aren't any really good naturally high C13 sources of ALA. Flaxseed, chia, all these like really high ALA containing seed oils are C3 pathway plants. There's a little bit of ALA in corn, but it's only about 1%. So, isolating ALA from corn maybe isn't the most efficient way to go about these studies. So we had to get a little creative. And one way to do this is simply to spike a very small amount of a post-switch ALA diet with C13 ALA. And by small amounts, we're talking no more than 0.05% of the total ALA as C13 ALA. Another way we've done this is to feed an algal DHA diet pre-switch. And what's nice about this is certain algal DHAs, they are often grown in the presence of a high C13 sugar source. So in the US, this is often going to be from corn, whether it's high fructose corn syrup or glucose, but also it could be in the presence of sugarcane sugars. So we have that high C13 background diet, and then we can switch to a lower C13 diet. So the first study I'm going to show you is using this approach where we spiked post-switch. And this is an ongoing study. Actually, the animal portion of the study is completed, and we're kind of wrapping up a lot of the data analysis in my lab. But here in this study, we had male and female mice fed either an ALA, EPA, or DHA diet pre-switched, and then there's switched onto essentially the exact same diet, but just with a tiny bit of C13 in it. And during this post-switch period, at multiple different time points, we'll collect blood, tissues. All throughout, we're recording a dietary intake as well. And that becomes important later when I show you some percent conversion values. And then we can analyze the fatty acids for C13 analysis and fatty acid concentrations. So just briefly, I'll show you sort of an overall summary. What we did show, liver DHA levels being higher in ALA and EPA-fed females. I'm not going to get into the sex specific aspects of this. It's a whole other presentation for another day, but we are seeing expected sex differences in the liver. However, these differences, we can't say whether or not these are actually due to changes in synthesis or differences in synthesis rates between males and females. So we really need this C13 analysis to determine synthesis and turnover rates. And because of the nature of the talk today, I'm going to really focus in now on the ALA-fed animals, as this is our primary plant N3 PUFA source that's the most abundant in our diet. So I'll focus on this and show here on the y-axis, we have our actual synthesis rates for plasma. There is no difference between males and females. In the liver, those differences in concentrations are being seen in our synthesis rates. So females had a higher liver DHA synthesis rate than males. And when looking at whole body synthesis rates, and just to be clear, when I say whole body, this is the body minus liver, minus blood, minus brain and heart, which we've collected separately, but at some point we are going to combine all of this as a real whole body analysis. But again, you can see no difference between males and females. And this is the big slide that I want you to really focus in on. Because we collected intake information on the mice, we know how much they ate per day. We were able to calculate percent of dietary ALA conversion to plasma DHA. So this is that value that I showed you earlier from human data, and we're showing at the same time less than 1% conversion, whether you're male or female in the mice. And the numbers are actually about 0.3 to 0.4%. So this is very much in line with what we expect. It also matches some previous rodent studies as well. But if we start looking at liver now, we have percent dietary ALA conversion to liver DHA. We're actually in the 6 to 10% range. So up to 10% of dietary ALA is actually being converted to liver DHA. And when we look at the whole body, and this is, a very large 20 to 30 times higher than what we're measuring in the plasma. With whole body, we're again in this sort of 9 to 10% of dietary ALA being converted to DHA in the whole body. And this is again 30 times higher than what we're measuring in the plasma. So the conclusion from this is that DHA synthesis rates from ALA are at least 50 times higher than previously estimated from plasma, at least in this model, this mouse model. And it doesn't necessarily mean that DHA synthesis rates aren't low still. They're just not nearly as low as what we might have thought previously. So if I switch gears here now and focus on a study where we fed algal DHA, and this is in collaboration with Nature Crops International. We wanted to use this diet switch model to assess plant-based omega-3 sources, particularly flaxseed oil and ahi flower oil. Ahi flower oil is really a very unique oil because it's about a third of its omega-3 PUFAs are actually in the form of sterodonic acid. Sterodonic acid is the first product of the N3 PUFA synthesis, resulting from the activity of the putative rate-limiting enzyme delta-6 desaturase. So the thought here is that by providing this SDA, we're bypassing the rate-limiting step and potentially synthesizing DHA at a faster rate. So we fed high C13 algal DHA pre-switch, switched to either a DHA diet, a flaxseed oil diet, or an ahi flower oil diet. And we can see the fatty acid compositions here. Most importantly here is we were able to manage to match the total N3 PUFA of the ahi flower oil with the flaxseed oil diet. We have our C13 signatures for the algal DHA being very enriched, fish DHA less enriched, and both ALA and SDA in the ahi flower, and ALA in the flaxseed as less enriched. So we get these decay curves, and just visually, if we just look at it, the orange ahi flower group looks to be on a steeper, more rapid rate of change than the flaxseed oil group, giving us the potential to detect these differences in DHA metabolism. The plasma half-lives actually appear to match the liver half-lives quite nicely, and that's important for potential translation to human work. The one limitation of this one is this high DHA background diet could artificially raise DHA levels during the post-switch period. So what we see, tissue DHA metabolism from ahi flower oil is not different from DHA. We have our half-lives here in plasma, in liver, and in brain. And in each case, the half-life for DHA is faster than from flaxseed oil, but not from ahi flower oil. And conversely, we could say the same about comparing ahi flower to flaxseed. And when we look at turnover rates or synthesis rates, we see, particularly in the tissues with liver and again with brain, the synthesis rates appear to not be statistically different between ahi flower oil and DHA. So flaxseed oil, as a result, is significantly different than DHA by all measures here. And as a result, tissue DHA turnover from dietary ahi flower is not different than from dietary DHA. So to start wrapping this up, just talk a little bit about some ongoing and future applications, continuing to work with nature crops to look at the role of a high N6 PUFA diet on DHA synthesis rates, with the idea that ahi flower might be able to avoid some of this competition between the two sides of the pathway. I'm also looking at liver-specific knockout models to see how there might be tissue-specific compensation for impaired DHA synthesis by the liver. And then we're now developing a study to translate these models into humans. So just to summarize, compound-specific isotope analysis is a powerful tool for investigating diet and metabolism, particularly being able to provide synthesis rates and not just percent conversion. We've found that synthesis rates from ALA are much higher than previously reported, and that plant N3 PUFA dietary sources have the potential to turn over tissue DHA rates at similar rates to DHA supplementation. Of course, much more work is needed for this, and I'd just like to acknowledge my lab, number of funding sources for the work, and the BASNet lab, who we continue to collaborate with. Thank you very much.
Thank you very much, Dr. Adam. And as mentioned earlier, we will have time for question and answer at the end of the two talks. I want to encourage you, please use the Q&A chat box in order to, not chat box, the Q&A box in order to capture your questions. Again, thank you, Dr. Matherel, for the really inspiring and eye-opening talk. And now we move on to Greg.
Hello, everyone.
The floor is yours.
Thank you. Hello, everyone. I'm Greg Cumberford, and I'm not yet a doctor, even though Rinat introduced me as one, but I've had the distinct pleasure for many, many years, more than 30 years, of working with many PhDs in the whole botanical wellness field. My whole career has been primarily in the industry side of botanical research and authentication. And I've been with Nature's Crops as Vice President for Science and Regulatory for just over 10 years now. And I really want to give thanks to first of all, the AOCS for sponsoring this talk, but specifically to Dr. Matherel, to Richard Bazinet, to Ken Stark, and the many researchers who have made it possible to elucidate some of these activities Going back in some cases 20 or 30 years, this research has been going on. Other researchers like Dr. Bill Harris and Jay Whalen, and even going back to Professor Keith Coupland in the UK and Michel Guichardon in France. There's been a tremendous amount of work that's been done. And I'm going to provide you a kind of health science summary that focuses in on what we feel are understudied omega-3, sterodonic acid, and icosahexatetranoic acid. And as Adam's presentation really showed, how there really is a way that plant-based sources can supply a more rich and diverse array of these anti-inflammatory oxylipin substrates, and actually biosynthesized to EPA and DHA with remarkable efficiency. So away we go. I'm just going to rehash a little bit from Adam's presentation. When we talk about classical omega-3 metabolism, we tend to think in a very sort of linear and schematic way. And so we see this classical, vertical pillar of the omega-3 polyunsaturated fatty acids, and sterodonic acid and icosahexatetranoic acid tend to just be seen as these sort of transitional fatty acids, and their main sort of role or value is only in how they can help efficient conversion to EPA. And it's sort of become part of that same dogma that Adam talked about, that exogenous preformed EPA and DHA sources are sort of considered better because they raise the omega-3 status more quickly or more efficiently. And in some way that makes preformed DHA the best omega-3 form because it raises that omega-3 status most efficiently, forms newly recognized D-series SPM metabolites, and still many people believe that there is a kind of retroconversion benefit, which Adam has described is really not the case. And for their part, the omega-6 side, essential linoleic acid is important. It's essential, but it's overabundant because of its prevalence in so many commodity industrial vegetable oils. So it's never considered deficient in the diet. And for its part, anti-inflammatory gamma linolenic acid has recognized benefits and efficacies, but they're kind of in their own world. And this of course, is correct, but it really doesn't tell the full story. A more holistic, and I would say cutting edge view, is one that really recognizes that there's this whole family of omega-3 and omega-6 fatty acids that start out with the essential ALA and linoleic acid, but which then presents to our cells in a much more holistic way, a much more complex and nuanced way, where these fatty acids are all serving in functional roles, and they're all providing substrates for these cyclooxygenase and lipoxygenase enzymes that are then producing these important signaling mediators that are supporting anti-inflammatory effects in many ways, sometimes very necessary and important pro-inflammatory effects. But it's just a more holistic view, and we'll bear that in mind as I go through my presentation. So Adam has basically just presented the significance of this slide, and in fact, he and his team are credited with demonstrating things like the fact that you can see an increase in plasma EPA following DHA supplementation, and that this is not occurring because of retroconversion, but instead slower metabolism and/or inhibition, as he described. But his and other teams have shown that substantial amounts of supplemental EPA are biosynthesized right through to DHA, despite no increase in circulating DHA beyond a kind of homeostasis in key tissues, especially the brain. And so it's important to recognize that when we put a lot of emphasis or a lot of focus on preformed DHA consumption, that's actually known to inhibit fat biosynthesis in the liver. And there's drug forms that are actually prescribed to lower triglycerides. There's this kind of view of plant-based precursors as being sort of surrogates for EPA and DHA. You know, they've certainly deservedly over many decades gained widespread scientific support for their role in various health issues that we all confront as we age. But we contend that their use is often prescribed in a sense, in a more drug-like way that is addressing more crisis issues once these disease factors have already set in. And you know, many diseases, in fact, do have chronic inflammation at root, but they could have been ameliorated much earlier if a healthier, more balanced plant-based diet had been employed in the first place. And humans and all mammals are biologically evolved to form all the tissue EPA and DHA necessary for optimal wellness, largely from plant-based diets. There's actually a recent 2022 literature review from FARI's Professor Ale Salavila that's showing how, you know, complementary dietary approaches from both ALA plant-based sources with EPA and DHA sources are optimal for more cardiovascular and cognitive functional outcomes. And I think it's important to recognize in this year, 2023, that wild sources are not going to be able to sustainably meet recognized dietary global intake needs, whereas plant-based, truly food-friendly dietary formats are going to need to be developed to meet the global need. Our industry's Omega-3 trade group, GOED, has recognized this fact as well in terms of how we cannot keep relying on the oceans. So it does put a premium on developing other plant-based sources. So let's take a little bit of a deeper look into Omega-3 SDA and ETA and a little bit into Omega-6 GLA. So sterodonic acid is considered that post-ELA, pro-EPA, Omega-3 polyunsaturated fatty acid. It's plant-based. It only occurs significantly in plants, although there are some marine sources like krill that contain small amounts of sterodonic acid. It's found in the seeds of a number of plant species in the borage family, of which ahi flower is one. As Adam noted, it's this sort of immediate byproduct in the conversion of ALA downward into longer chain omega-3 fatty acids. And it does bypass that delta-6 desaturase rate limiting step. So we know now that consuming oils rich in SDA results in far higher enrichment of not only EPA, but also some of these other important omega-3 fatty acids, ETA and DPA, but of course not DHA, as Adam showed. One thing that's more newly discovered, which was done in a human clinical trial carried out in Canada through the University of Moncton and Professor Marc Serret and Natalie Lafort and their team, was the discovery that SDA is actually triggering an interleukin-10 upregulation effect. Interleukin-10 is the sort of anti-inflammatory cytokine that our bodies naturally produce during immune challenges. Its levels in macrophages can be significantly impacted by a rich sterodonic acid source like ahi flower. In the human clinical trial that they published back in 2017, they noticed a plus 40% increase compared to baseline in immune macrophage cells. And they noted that the abundance of interleukin-10 can predict the severity of several human diseases with various kinds of pro-inflammatory etiologies. And they were good to point out that this was the first investigation of its kind that showed this sort of immune modulating response following the consumption of ahi flower oil. So they're suggesting that it could share immune modulating properties that are typically associated with marine oils. Then there was another study that came out in 2017, which was an in vitro trial that showed that ceradonic acid has comparable cell signaling as DHA in terms of specific inflammation cell signaling pathways. It inhibited nitric oxide produced from inducible nitric oxide synthase in macrophages comparably with DHA. SDA and DHA similarly inhibited LPS-stimulated NF-kappa B expression in macrophages. SDA and DHA comparably inhibited MAPK phosphorylation. And so they're positing it as an underlying mechanism of SDA's anti-inflammatory effects. And they said that SDA may be used as an alternative to marine-based fatty acids for preventing inflammatory diseases, and that is as effective as DHA against inflammatory response in LPS-stimulated macrophages. So that's pretty valuable and interesting, especially in light of what Adam just shared with us in terms of efficient DHA biosynthesis. Then there was also a study that came out in 2017 looking at endpoints in terms of why we think it's important to increase DHA either through supplementation or biosynthesis. This was a study carried out at the University of Illinois, where they found that in healthy seniors, it wasn't a dietary intervention study, it was actually looking at their MRI brain scans, and they were looking at correlations to fluid intelligence, and they found that circulating ALA and SDA and the metabolite ETE, eicosapentaenoic acid, were what mediated fluid intelligence and total gray matter and frontal neocortex brain integrity in healthy seniors, but not circulating EPA or DHA levels. And then coming back to the team that Adam is part of with Richard Bazinet, they did an important study that was asking how much DHA does the adult brain use? And they were able to show that this is actually a far lower amount than we might think, at the level of single milligrams per day, rather than tens or hundreds or gram levels of DHA that's being consumed. And that's an important, I think, consideration. And we're always also clear to state that this is about adult healthy brains, not for well medically indicated recognized uses in prenatal and neonatal nutrition, where one is building a brain instead of just maintaining optimal brain function. And this just touches on a topic which could be an entire presentation in itself, but which is to say that when we're talking about oxylipins, there's a growing body of important research that's recognizing that these oxygenated lipid messengers are really the main mediators of the dietary omega-3 and omega-6 PUFAs that we take in to our bodies. They're formed by these cytooxygenase and lipoxygenase and cytochrome P450 pathways, and they're resulting in the formation of all these different arrays of molecules that really are linked back to the desirable pro and in some cases anti-inflammatory effects. And the reason that we bring this up is because although they have both pro and anti-inflammatory activity, they generally tend to increase in a dose-response manner with increased EPA and DHA intakes and or status in the body. So one of them is this ETA that is fairly newly recognized for its status as a new oxylipin source. Just in 2018, researchers in Canada identified a 5-LOX metabolite with anti-inflammatory activity, showing inhibition of neutrophil leukotrienes and neutrophil chemotaxis, leukotriene B4 receptor agonist activity. And they said that the metabolites of icosahexatetranoic acid could possibly possess anti-inflammatory or pro-resolving activities analogous to the well-described EPA, DPA, and DHA resolvins, protectins, and maricins. And that's important in our considerations around SDA and its effects, because dietary SDA is going to be the most efficient way to boost circulating ETA levels as well. What about GLA for its part? GLA is not so common in the diet. It's typically found in only a few botanically derived sources. But it has a long history of literature of supporting anti-inflammatory benefits to things like women's reproductive health and hormonal balance and skin health. It too skips that same delta-6 desaturase enzyme rate limiting step and is readily converted to its metabolite dihomo-gamma-linolenic acid. And it's the only one thus far on the omega-6 family that's positively associated with increased longevity. It is also a generator of DGLA, increasing DGLA levels, which have their own recognized anti-inflammatory oxylipins. So in the one of the human dietary studies with ahi flower oil, DGLA was significantly increased by 16%, where in that same study, flax oil, it decreased DGLA synthesis and levels by 16%. So when you stack those two together, going back to that sort of more holistic view of what's happening at the cellular interface, we see that both ETA and DGLA are serving as these important substrates for anti-inflammatory oxylipins. And it's significant because conventional EPA DHA sources are not going to achieve that. They're downstream essentially from that in that sort of balanced way. And for its part, ahi flower oil will be the richest dietary combined source for achieving upregulation of both ETA and DGLA. And that's in complement to the others that are downstream that that the research that Adam's team has been working on shows is happening with ahi flower consumption. Compositionally, ahi flower, as Adam noted, it's a very rich source of sterodonic acid. In fact, it's the highest naturally occurring source of sterodonic acid, but it is also an excellent source of alpha-linolenic acid, about 75% of that of, say, flax or chia. It is a source of linoleic acid, but relatively low. And it is a good source of gamma-linolenic acid, as well as 10% oleic acid, like we might see in oleic acid containing oils like olive oil. So overall, it's the richest omega-369 dietary source available from a plant at over 90%. So that's just it from a triglyceride compositional standpoint. The first human clinical trial, as I alluded to earlier, showed that dietary ahi flower oil increases circulating EPA in 4 different circulating cells, up to about 4 times more than dietary flaxseed oil. That's important for a lot of functions, including cardiovascular and even brain DHA formation. This is work that Adam's team has done. This recapitulates one of the graphs that he showed, but it is showing that in just the first 14 days of that dietary time trial, looking at plasma and liver DHA biosynthesis, you're seeing how tightly separated the ahi flour is in the red from flax in the green, but how close it is to the marine DHA-fed animals. And so it comes out to be about 80 to 90% as efficient in that respect. I'm mindful of the time, so I'm going to keep going. They also, in the same research, looked at levels, and the liver and brain DHA levels didn't vary statistically significantly in any of these cohorts. And that's important because it sort of reaffirms that the mammal brain doesn't really need to take in preformed DHA in the diet to maintain adequate tissue DHA levels among adult animals. And again, we stress that that's different than when we're talking about supporting prenatal and neonatal nutrition. And then the last ahi flour study that I wanted to draw attention to is, once again, a study. This was in mice in TPN. So this is intravenous uses of omega-3 fatty acids to support cellular function when the whole gastrointestinal tract is not available. And one of the notable findings from this study in mice, where they compared an ahi flour-based emulsion to either fish or soy-based emulsions, was the notable upregulation of interleukin-10 in various tissues, the liver, the muscle, and the white adipose. The interleukin-10 was significantly higher, but also the interleukin-6 to interleukin-10 ratio was significantly lower in those tissues. And they credited the ahi flour-based emulsion with having remarkable anti-inflammatory, insulin sensitizing, and immunity enhancing properties, acting as a kind of immunonutrition. And this was to overcome some of the noted challenges that exist currently with either the fish or soy-based intravenous emulsions. So thinking of this as a kind of paradigm approach, if it's possible to shift to a truly more preventive approach than a reactionary approach in terms of wellness, the research that is coming forward really suggests that a plant-based source like ahi flour can be part of that, and we can be thinking more proactively across the whole diet of ways that we can achieve those benefits, as opposed to looking at thinking that we have to respond later in life with only preformed EPA and DHA sources. The benefits of that, of achieving better balance in terms of omega-3 status and omega-3 intakes, I think is undisputed. But the need to do it entirely from preformed sources, I think, raises a lot of questions. And the research that was described on this presentation is starting to indicate why that is the case. And then shifting from the cell out to, epidemiological scale, this is a recent, fairly recently published October 21 NHANES study that was looking at CVD and cancer prevention, and looking specifically at omega-3 status and what were the sort of broad population-based correlations and associations. And what they found was that higher serum levels, not only of EPA and DHA, but also linoleic acid and alpha-linolenic acid, showed robust inverse associations with all-cause mortality, not just EPA and DHA. They also saw that higher serum EPA, but not DHA, correlated with lower CVD death risk. And they found that only higher serum ALA correlated with lower cancer death risk, not linoleic acid, EPA, or DHA. And they concluded quite logically that suggestions should be taken to increase marine-derived and plant-derived omega-3 fatty acid intakes to prevent not only all-cause mortality risks, but also other cardiovascular cancer mortality risks. So that's that balanced approach driven by allowing the legitimacy and the role of omega-3 plant-based precursors to go alongside the omega-3 marine sources that I think achieves a more sort of sustainable system-wide outcome. And I guess really in conclusion, you know, it is in the end all about how are we going to achieve greater balance? How are we going to take full advantage of food formats that are scalable and sustainable over time for achieving effective omega-3 status in the body. And it shows that that there are sources like ahi flour that can achieve that, presenting the body with a more balanced and adaptive cellular platform to support this kind of overall anti-inflammatory response than you might see if you were only looking at EPA and DHA sources. And that also plant-derived omega-369 dietary oils bypass this key rate limiting delta-6 desaturase step and do provide an efficient and optimal platform from which the body can address both acute and more sort of chronic inflammation states. So as a proud sort of person who's been developing and making available ahi flour oil in the world, we really feel like its time is coming where ahi flour is really well suited to helping people achieve those broader cellular wellness, but also ultimately epidemiologically verifiable outcomes. And that is my presentation. Really happy to take on questions, and I'm also happy, given the time limits, to address anyone's questions one-on-one. My presentation will be made available subsequently to this talk. You're on mute, Rinat.
Thank you for this. Thank you for this interesting and inspiring talk. And indeed, we are open for questions and for the Q&A part. We have a few questions. Let's try to cover them all. The first question is for Adam, and I will try to summarize it. There is a question if it is possible to process the studies that you've done also with high oleic palm olein, or perhaps also with other oils. And if yes, if you can share some contact with us, but really the question is, can we repeat those also with other oils?
Yeah, the nice thing about this modeling is you can really do it with anything, especially knowing, you know, with palm oil, it's coming from a C3 plant source. There's not really a great, there are other obviously C4 sources of oleic or palmitic acid, but the simplest way might be to do that very small spiking of C13 oleate or palmitate in the study. And really these can be applied to anything that you could use, do it for sugars, amino acids, anything that's being metabolized to downstream products is, it's very possible to do with this methodology.
Oh, thank you. We have a few questions for you, Greg. Mm-hmm. One question is, how much ahi flour is recommended for a daily dose to considering that it is a novel food? Why is it not allowed to give ahi flour to young children in food supplements? And
then I'll address the next question as well around the conversion factor. Mm-hmm. So when we're looking at Ahiflower from a maintenance intake standpoint, we're looking at 2 to 2.5 grams per day. And that ties to recognized peer-reviewed published research that has looked at these ALA and SDA conversions through to EPA. And that usually sits at around 30 to 40% efficiency. So if your aim is to achieve World Health Organization EPA accrual levels that are around 250 milligrams EPA, then that sort of pro-EPA equivalency translates to 2 to 2.5 grams of Ahiflower per day. It's only in the EU that there's a recommendation around Ahiflower in young children. That's not the case in the US or Canada or other jurisdictions. And I think it just has to do with an abundance of caution approach that was taken originally during the preparation of the dossier. There's no fatty acid that's in Ahiflower oil that isn't abundant in many, many, many other vegetable seed oils. But that was just a regulatory outcome that came out about 9 years ago when EFSA was originally considering Ahiflower oil.
Okay. Thank you. And we have another question from the audience. Has Ahiflower oil been approved to use in dogs and cats?
It is used in dogs. We don't recommend Ahiflower for cats because there's a different hepatic enzyme capacity that cats have for converting linoleic acid and gamma-linolenic acid. So it is used in canine supplementation here in North America, and we're in process in other jurisdictions. In the EU, it's already considered to be accepted as what they call an animal feed additive. So in most jurisdictions, yes, for dogs and also for horses. There's quite a lot of equine Ahiflower already in the world through Purina in the USA and Pavo over in the EU.
Thank you, Greg. Another question, is there any approved health claim on Ahiflower oil, such as high in SDA?
You can make compositional claims like that, and you can make other ALA-related health claims because ALA is recognized in the USA, Canada, and the EU, for example, as having conferable health claims. But you have to go with what your jurisdiction says in terms of ALA content. And over time, we expect that we will develop Ahiflower SDA-based functional health claims pending the outcomes of current human clinical trials that are happening around the world right now.
And if we have time for one last question, the DHA resultant in the rodent models is similar from fish or plant sources, from what the person who's asking the question is understanding. Assume blood levels were measured. What about tissue? Could there be levels elsewhere in the body?
Yeah, so I'll apologize now because I think I started going through those slides a little quick, realizing I was getting short on time. So I probably didn't spend as much time as I wanted to on them, but we have plasma, liver, brain, and just this past week, adipose as well. And they're all showing, if not identical, very similar results. So we didn't collect in that study whole body, so we're not going to have that whole body assessment, but certainly every tissue we've looked at so far is very similar.
Cool. Thank you very much. I would like to be sensitive to the time, so I want to thank our speakers, and I also want to thank all of you for attending this webinar. I'm hoping to see you in Denver at the end of the month for the AOCS annual meeting, but if not, Please stay tuned. We will have more webinars, and we hope to see you soon in the future.
Thank
Bye, everyone.
you.
Thank you.
Thank you, Rinat.
Bye-bye.
Bye, everyone.